they target tropical insects. Okay, yes, it's going to be recorded. And why is that? Because, well, there is a very high alpha diversity, local diversity of tropical insect in any place. The reference libraries for those, insect where we can really get the sequence from are really scarce. And then, of course, it's very expensive just to build those reference libraries, especially when you have high alpha diversity. In addition to that, the taxonomic knowledge might be pretty much, pretty much basically restricted. We have also many undescribed species, which are known only by the barcode, for example. The ecological information is also fairly restricted. That's important because the geographic, information, the distribution, geographical distribution is often limited to absent. So you can say that those represent model organisms to really test the limits of metabarcoding, and that's what we're interested in. But also maybe the organizer of this meeting are really also a group of entomologists, and I would say also that insects are cool, really, and that we need more people, to study them with or without metabarcoding. And we hope that, those 2 days might actually stimulate students to start again some studies, either with metabarcoding or without metabarcoding. So just to explain how the meeting is organized, we're going to have 16 presentations, that's 8 per day. And then what we want to do is to, after each 4 presentations, we're going to have a discussion, sort of basically question for the speakers, and then general, discussion about the presentation, but also about, different topics, which I think will be better than to have basically, okay, sorry about some of the notes appearing on the screen here. So we'll have, discussions to sort of recap a bit what we might have. That's good. So everybody is just listening to us, so that's excellent. So we have basically discussion about those different presentations, but also more general. We're going to have coffee break. They're pretty long, like half an hour, but they will, allow us to recap any kind of time that we might, we might take longer with the presentation. The coffee break are going to be upstairs, and no food or drink are allowed in the auditorium. The lunch break, so yeah, I know this is tough, but try to keep your coffee juice up. So the lunch break, unfortunately, the cafeteria, the Stride, the Switzerland cafeteria is closed, is temporarily closed. So you can have, if you have your own lunch, you can basically sit in a table of the cafeteria, and then I think we send maps with some places where you can get lunch nearby. Also, maybe for the students who might be attending presentially, I think if you need a certificate of participation, just drop me a line or to Eduardo Navarro, and then we'll send you the certificate. Before I actually introduce the SENACYT project, the SENACYT metabarcoding project, I would like to say a few words about our Stray Arthropod program, because this is all sort of embedded in this. And this is a program that's been monitoring insects on Barro Colorado Island, so in the middle of the canal here in Panama, for 16 years. We have 23 insect group that we're monitoring, so pretty much different from butterflies to termites and so on. So far, we have accumulated something like 0.8 million of insect records. This is all in a public database. We have we're monitoring about 2,500 species. We have a collection with 80,000 specimens on 12,000 sequences with the voucher in those collections. So that's what is important for metabarcoding. This is supported by something like about 100 gigabytes of data, which were processed by this for this project, basically. But now we're going into next generation monitoring, not only with the DNA metabarcoding, but also with bioacoustic. And then every month, for example, we're generating 1 terabyte of sound data. And every week we have 10 systems on the island, which are basically generating 21,000 insect pictures who are basically processed by artificial intelligence. And we have an international collaboration on many publications and so on. So this is sort of the organigram of the different sort of project that we have so far with this program. So you can see that, most of the this is related to the traditional monitoring, but with the new, technologies, I say. But we're also doing a lot of other stuff which might be related to climate change or not related to climate change. The idea of the monitoring, and that's important, is just to basically use either clades or functional group, to group them and then just to see the the patterns over time. We have an estimate of about 25,000 species, arthropod species on the island. So we're monitoring, we're monitoring about less than or about 10% of those species if our estimates are correct. And why is it that basically we're doing all this monitoring on BCI, on Barra Colorado Island? It's because it's safe from deforestation and pollution, and then that the program is building all this reference, library of insect with the specimen, with the sound, with the images, the sequences for those 16 years. So that's very important for this. And we have access, of course, to excellent meteorological and vegetation record that we can basically put in relation with insect data. And of course, there is excellent logistic and safe working condition. So here, for us, what is interesting is that we can study those insects in an environment which is really safe of the main anthropogenic stressor, but not of climate change. And that's what we're interested in. That's the 23 taxa that we're monitoring on the island with different protocols. And that's important because they basically that will condition what kind of DNA reference library we have. So you can note that there are lots of Lepidoptera, like moth and butterflies, and for example, a few beetle groups which are being monitored. Just to say one or two things about the result. We have lots of different response to climate change, and it's not only between the insect group, the insect taxa, but also between the species within a certain taxa. Here, this is an example of tiger moth, Actine, where we can clearly see that some species are in decline, others are stable, and actually many species for the time being are actually increasing. So the prospect for this STRIDE program, I have no doubt that this is the longest running and the most complete annual census of tropical insect communities in the world. And I think this should be protected at any time. We were funded by Forest Geo, which depend on the Smithsonian until 23, but then they switched their interest to tree monitoring and to carbon cycling, and they cut all our funding. We got modest bridge funding for this year at STRIDE, and nothing is really decided for next year, so we don't know basically what's going to happen on all of this arthropod research on BCI. If you want to help or discuss this matter, you can just drop me, a message. But getting really into this metabarcoding, the SENACYT, what we call the SENACYT metabarcoding project on BCI, we have basically on BCI 50-hectare plots from where we actually had 25 small plots. This is only 20 by 20 meters. And here, all the composition of the trees, of the plant, of the liana are known. And we actually process insect sample with 7 different, protocols. On the next talk by Daniel Souto-Villaros is going to review these methods on the difference between those methods. And so basically we have vegetation data, we have also ground LiDAR data for all those different plots, so a lot of, ecological information about the plot. We actually took in total 350 samples. That's basically 7 protocols across 20 plots on 2 seasons, dry and wet season, so that we pooled all those samples together to this insect soup to do the metabarcoding analysis. So the objective of those data, that's first, and I think Daniel is going to talk about this in the next talk, is just a selection of the best protocol to detect the beans, because that's, for insects, it's very different from trees. Like trees, you have just basically one protocol and that's it. But, you have as many insect protocols that there are, insect group. So that's, one thing to consider. We are really interested in the fine, spatial distribution of those insects in the plot, in the forest. So that is looking at the relation between the vegetation, that's not only the species richness of the trees, but also the liana. We're interested in the, the relation with the composition, what you have in terms of vegetation in the plot, for example, to see whether any kind of tree species might have some predominant effect on the arthropod fauna. We're going to look at the, the local plant phylogeny versus the insect. And we're interested also of the effect of the forest structure. So that's the LiDAR data. That's basically the leaf area, the total leaf area, the what is called the CVD, the coarse woody debris. That's the dead wood, basically the dead standing or lying wood in the plant, and then the canopy upness, of course, on other variable. But we also, with those data, we can also look at the overlap between the soil fauna and all the fauna which is above ground, and that's interesting. In the end, of course, we're interested to know how many species we have really on the island, and this is maybe a first. This will represent the first hint. What is interesting here is that we can do some calibration with all the insect group that we're being monitoring, because we basically know, or we have pretty good estimate of how many species we have on the island for those groups. We also doing bioacoustic at all of those, 25 plots, so that basically for each of those, 25, quadrat, we have metabarcoding data, some data, vegetation data on the forest structure. So that's going to be extremely interesting. So I'm going now just to have sort of 4 slides, which actually summarize some of the primary data. As you can imagine, you can see that, from different angles. Again, my colleagues, Daniel Souto-Villaros, will talk about the protocols, and tomorrow, Eduardo Navarro is going to talk about the relation between, the bean detected on the vegetation. All together, we got about 10,000 beans, so barcode index number, more or less, species, because we've been extremely, I would say, cautious about the filtration of the data, and then we have very conservative estimate. This is a very conservative estimate, which probably might be decreased also a little bit. That's over the 25 plots, so they are only 20 by 20, which means that in 1 hectare of forest, you have a distributed hectare of forest, you have at least 10,000 species, I would say, which is quite remarkable. But if we look at, for example, the distribution between the orders, the mine order, the 6 mine orders, we see that the diptera, the flies, are very sort of important in the number of beans detected over those 25 plots. And the Lepidoptera, the moth and butterflies, also pretty important. And then in ranking, the beetle are third, on the Hymenoptera only fourth, and then the bugs, the Hymenoptera have wasp and bees, and then the bugs, Hemiptera, fifth, with the spiders being last in this list. I mean, here it's interesting because you could look at the number of described species, in the world, and the ranking is pretty much different, with the Coleoptera normally to have the most described species as an order, then the Diptera, then the Lepidoptera, and only in fourth, place the wasp, the wasp on the bees. But many. There's many sort of hints that people disagree with this. There is, of course, a bias in the, the description of those taxa, and probably that either the Hymenoptera or even the Diptera actually have more species than beetle, which means that basically we should have any of those three, the beetle, the flies, or the wasps should be the sort of the main order that we should actually, in a tropical rainforest, we expect to see. But that's not the case. So I think here it's probably in part, influenced by the reference library that we're using, because as I said, for example, we have a lot of data for the moth, especially on the butterfly. There is 600 species of butterflies on the island. You have only 60 species of butterflies only in the whole of the United Kingdom. So another magnitude more of species on this island. And basically the beetle are not very well represented in our sample. So basically, we might have a problem with the, I would say, overall representation of the data. And then that's, I think, something that needs to be, considered. And Jeremy DeWard is going later, I think this morning or this afternoon, just to talk about this, about the reference libraries. But I mean, what you can argue is that you could still make a relative comparison, with, for example, different location. In the case of the study, we're interested to compare those 25 plots and then to see where the arthropods are. So those sort of a relative comparison are still valid, because this is what basically we know, and this is the best that we know. Another, possibilities that here we only consider barcode index number, BINs, and then we have also lots of OTU, which we believe do not qualify as being as a species, if you want, because they have no voucher. We don't know what they are, and then they actually appear. The sequence length is extremely low and so on. But you could maybe argue that if we put those all together, instead of considering only the bins, maybe you might have something which, might be closer to, let's say, the reality. So that's one thing to consider. If we look now at the distribution of those, the same orders, for the 25 plots here, basically, overall, we have about 2,000 species. So it's quite interesting to know that, in a small, plot, like by 20 by 20 meter, you can have already 2,000 species of arthropods. That's, quite interesting in itself. But what you can see is that there is a relative sort of uniformity with, again, it's the number of bins in each of the plot. So there is not too many differences, between the plot. And this can be explained by the fact that we wanted to have the data to be representative of a forest, which is sort of an old growth forest, like we avoided, any kind of large forest gap, anything. So the structure of the plot is basically, and the composition should be, reasonably similar. So I think it's not too surprising to see that we don't have huge difference. We're not comparing, for example, plantation with, primary forest and so on. So that's one thing to consider. But here, the challenge is that this is just the number of bins per plot, so the species weakness per plot, and you can genuinely ask whether this is a good indicator. I mean, if you look into all the entomological literature, mostly people will advise not to consider species weakness per se, but at least the diversity, so modulated by the abundance, or even the abundance itself on different categories. So that's another challenge, because our sort of immediate data that we can get with metabarcoding is presence absence. We're going to have some talk who actually going to talk about this, and that's clearly another challenge. Now, if we look at the occurrence, so the occurrence or the prevalence, if we want, like for those 350 samples, we actually took 3 replicates just in the lab analysis. And so we have altogether, we have about 1,000 samples. So you can look now at the occurrence of those bins, in each of the sampling method across the season, the dry or wet season. And you could basically have the same sort of distribution here. It's slightly different. Like I actually very quickly assign each of those different, bin, those 10,000 bin to different, feeding guilds. Like, for example, leaf chewing, the leaf chewing in green here are pretty well represented. The scavenging fauna, that's all, the fauna which is associated with the dead wood, with the litter, with decaying matter, and so on here. And that's basically the two, especially the scavenging fauna and the leaf chewing, insects are basically the dominant feeding guild. Again, you could say maybe this is also influenced by, I would say, reference libraries. For example, the parasitoid is just below here, the leaf chewing here. That should be, this is all what we call, we entomologists, Hymenoptera parasitica. This is extremely diverse, but very few people are working with this, and certainly the reference library are not there just to see this. So again, I think here we have certainly an influence from the reference library. I mean, the other challenge again is, well, is this prevalence, this occurrence, sort of more informative than simply counting the number of species? I would say yes. I think it's not exactly the presence. It's again presence absent data. It's sort of closest to what we have to abundance in those. So that's interesting. But there might be other methods to do that. And then if we go a bit further, like if we look at again the prevalence, so the percentage, occurrence in those, 1,000 sample, we can see what kind of sort of the most common species we have here. We have the winner is a grillid, like that's a cricket. Then we have wood lice, we have columbola, we have earwings, we have lots of cockroach, and then there are lots of common beans like bark beetle, columbola, mites, termites, on rove beetle, all those species which are very well represented in the sample, they are basically scavenging fauna. Maybe the earwing is predatory here. So the question is, okay, this is just looking at the occurrence, the prevalence in the in the sample, but you have other sort of ways of expressing this. We can look at the number of reads, the relative read abundance, which is often used, the number of sequences, the prevalence, or maybe some other variable of interest. And we're going to hear about this in other talks here. Is that related to biomass? Is that really representative? Is it really those species which are the most common species on the island? I would say what is a bit surprising here is that you don't have any leaf chewing or sap-sucking insect like herbivores, which sort of appear in this palmarès, in this list of most common species. So that's a bit surprising. One sort of, I would say, possible explanation is that our sampling, and I think Daniel will talk about this, targeted the understory of the forest, so the lower part of the forest, because by experience, we know that the canopy, you have different insects, and you need to have different protocol to sample the canopy, and we couldn't actually do all of those protocols. And of course, in the canopy or in the higher part of the forest, you will have more leaves, you have a higher leaf area, and then you will have also more herbivores. So that could be one possibility. I'm going to stop here. This is sort of the acknowledgement side. I think we are